and gentlemen, welcome to mediSars. And you know what we do on this show, right? This is where we talk about whatever that concerns medical science. Here we talk about anatomy, physiology, biochemistry, pathology, pharmacology, and all those medical sciences that you can mention. And then here we give you the opportunity to suggest those quite confusing topics and courses you don't really understand, okay? So that at the right time we break them down to the fine degree that you understand so well. We're going to start with a very fundamental concept in medical sciences, that cell structure and its function. But before we start with everything, you've got to subscribe, okay? So look down your video screen to your right and tap on the subscribe button, okay? When it is subscribed, it means that you fully subscribe to the channel. And also don't forget to hit the notification bell so you'll be notified on your phone whenever a new video is posted, okay? And if this video finally deserves it, please try and like as well. Let's get into it then. Okay guys, so now let's try to enter into the cell right now. So supposing you go to the laboratory and then you want to take a close look at a typical human cell, okay? For that matter, the laboratory technician assists you with a light microscope, and then you want to look into the cell and see the various parts that can be seen under the light microscope. So that's the question you're asking yourself right now. What are the various parts of the cell that you can possibly view under the light microscope? That's what we're going to start with right now, so that later on we deal with more detailed concepts, okay, concerning the cell structure and function. So let's get into it. The various parts of the cell that you can possibly view under the light microscope are three, okay? What are we saying? There are three basic parts of the cell that you can view under the light microscope. What are these? So under the light microscope, you can see the cell membrane, okay? Also, you can see the cytoplasm of the cell. Okay, under the light microscope, cytoplasm is visible, as well as the cell nucleus. Awesome. So guys, these are the three basic parts of the cell that you will see under the light microscope. Okay, so let's try to put this into diagram to know where we can find our cell membrane, where we can find our cell nucleus, and where possible we can find the cytoplasm. So this is going to be our cell nucleus, but I know the cell nucleus can either be placed at the center of the cell, but not always. Remember, some cells have got their nuclei shifted close to the base of their cell membrane. Because of that, we say that those kind of nuclei are termed as peripheral nucleus because they are shifted to the periphery of the cell. But in our illustration, we are considering a nucleus that is centrally located. So we call it a central nucleus. Fine. So obviously, this structure is our cell nucleus. Why is our cell membrane? Where can you find our cell membrane on this diagram you have on the board? You know from basic school that the outline of this diagram, okay, this cover of the cell is what you call the what? The cell membrane. Awesome. And then our cytoplasm is simply the space confined within the cell where the cellular organelles, including the nucleus itself, are found. Right? So this is going to be our cell cytoplasm. Awesome. So this is our cytoplasm. Take note of that. So guys, basically these are the three basic parts of the cell that you will see under the light microscope. I must tell you that sometimes, depending on the magnification power of your light microscope, you can see other cellular organelles. Okay, so aside cell nucleus, some cellular organelles can be seen. In fact, even all, but not as clear as the cell nucleus. You see them as densely distributed stains, okay, in the cytoplasm, densely distributed dots, as I'm illustrating the board right now, in the cytoplasm of your cell that you are viewing under the light microscope. But remember that not all magnification will allow you to see what? The cell organelles as even densely distributed dots in the cytoplasm. So take note of that. But one thing that you need to also bear in mind is that there is no way you'll be able to view any of these cellular parts unless you stain your cell. That tissue sample you place on top of the slide on your light microscope to view, you have to make sure that it's well and appropriately stained with a dye. And you know that at the pathology lab, especially, there are a lot of dyes that can be found in there, but the commonly used dye, in case you don't know, is what we call the hematoxylate and eosin dye. It's a one type of dye, okay? They are not two. Hematoxylate and eosin dye. That's what we normally use to stain our dye. We normally put it in short HNE dye. Awesome. So guys, you've got to stain your dye before you can see this part. So what you're going to do right now is that We're going to bring out these structural parts of the cell and make sure that we enter into them, know their detailed structural components and how these components function together to help the cell exhibit its functions. So on top of our list is the cell membrane structure. So we're going to look more into the cell membrane structure. Okay guys, now what is about the cell membrane that's making it so special? Okay, the cell membrane is actually a special structure around the cell. Why? Because it has all the 3 basic digestible macronutrients that we take in every day in our diet. Do you remember the various food sources, proteins, carbohydrates, lipids, you have the mineral salts, the vitamins and water, right? But the 3 macronutrients among these 6 are the proteins, lipids, and the carbohydrates, and they are all found in the cell membrane structure. So what are you trying to say? It's making it so special, right? You are saying that the cell membrane structure is made up of what? 3 basic macromolecules, termed as what? The proteins. In the cell membrane you can find some types of lipids. In the cell membrane you can find some traces of carbohydrates. Is that okay? Awesome. Take note of these. Now, it is important to know these macromolecules forming the structure of the cell membrane. But do you know the next question that we're going to ask ourselves right now? How much proteins are in there? How much lipids can be found in the cell membrane? How much carbohydrate? Because of that, it's going to lead us to talk about the percentage compositions of these macromolecules in the cell membrane structure. So how much proteins are in there? In the cell membrane structure, proteins cover up about 55% out of the overall structure of the cell membrane. Now, how about lipids? Lipids are taking up about 42%. Okay, so now because we are dealing with what percentage and it's over 100, 55 adding up 42 lipid component is giving us what? 97. So the remaining 3 now goes to what? The carbohydrate traces in the cell membrane structure. Is that okay? Now, after knowing these structural components, okay, the next thing that we're going to talk about is that because we are saying that these macromolecules are making up the entire chunk of the cell membrane, such that wherever that you pass in the cell membrane structure, you either find proteins, lipids, or carbohydrates. It means that they are served as the building blocks of the cell membrane structure, right? So in case we are trying to build a cell membrane right now, for which we are going to do later in this same lecture, what building blocks are going to serve as the foundation of the cell membrane? In other words, which of these macromolecules are going to serve as the building blocks for the foundation? Is that okay? Fine. And this is where I tell you that it is the lipid components of the cell membrane structure that are serving as the building blocks. Okay, so take note of that. They are serving as the building blocks for building the foundation of the cell membrane. Is that okay? Fine. So because of that, we will take the lipids in isolation and deal with it. Fine. But you know that on the surface of the earth, there are a lot of lipid molecules discovered. So what types of lipids are peculiar to the cell membrane structure? That is also important to get knowledge of. So now let's try to enter into it. What types of lipids are found in the cell membrane structure? In the cell membrane structure, there are basically 3 different types of lipids. What are they? Predominantly in there, you can find phospholipids, okay? Phospholipids. They are the common types of lipids that can be found in the cell membrane structure, okay? Later on, we know the different types of phospholipids that we have in the cell membrane. How much phospholipids can be found there in terms of percentage? So in terms of percentage, phospholipids are covering up about 25, let's use a different marker here, where it's not working. It is contributing about 25% out of the overall structure, lipid component of the cell membrane. Is that okay? Now, what about the other types of lipids that we have? There is this other type of lipid in the cell membrane structure that we normally mention in our environment. Sometimes we are restricted to reduce its intake. We call it cholesterol. So cholesterol are also serving as building blocks for our foundation of the cell membrane. How much cholesterol are in there? The lipid component of the cell membrane, they are taking up about 18%. Is that okay? Fine. Now, what is the last type of lipids found in the cell membrane structure? We call this type of lipids sphingolipids. How do you call them? Sphingolipids. They are taking the remaining one. Let's try to do a mathematics again, our calculation. We know that we have 25 adding up to 18, giving us what? 38. And the total is what? 42 lipid components in the cell membrane. So if you're having 38, how much left? 4. So the 4% goes what? The phospholipid type of lipids in the cell membrane structure. Okay guys, so now we now have our lipid building blocks for the foundation of our cool cell membrane structure available to what? Our phospholipid structures, our cholesterol, and then our phospholipids. Okay, we know the percentage compositions of these lipid building blocks as well. But you now come back to remind ourselves of what we started. We said that it is these building blocks, okay, the lipid building blocks are the ones going to serve as our resources for building the foundation, okay, of the cell membrane structure, within which we can find all other structures like the protein, the carbohydrate. Get it? So now that we have them available, we're going to start with how they typically look like. Okay, you know, from this time forward, we are going to build our cell membrane structure, the typical human cell membrane structure. Because of that, we have to have our foundation on board so that we can know what we can adapt to it. Talking about the proteins and the carbohydrate. So you're going to permit me to bring this part of our board so that we start looking at the typical structures of the phospholipid, the cholesterol, and the sphingolipid. Okay guys, so now we have our lipid building blocks for the cell membrane's foundation available, right? Yes, we said that the phospholipids, the cholesterol, and the sphingolipids, they are serving as the resources for building the foundation, okay? So what we're going to do right now is that we're going to enter into their detailed formation, their detailed structure, okay? So that we know how they typically look like. And then later on, we know how we can derive simple and much more smaller structures out of them so that you can use those structures to represent them wherever that you're trying to draw them. Is that okay? So let's get into it. We're going to pick the phospholipid structure first. So how does phospholipid typically look like? How does phospholipid typically look like? In the structure of phospholipid, there is this backbone called glycerol. And what is this glycerol at all? Glycerol is actually a 3-carbon compound. It has 3 carbons, and these carbons are binding to 3 hydroxide ions or hydroxyl groups, and then the bonds around the carbons are closed up by 5 protons. So what we have on the board right now is a glycerol backbone of our phospholipid structure. Fine. Now, because we are saying that this serves as our backbone, it means that that's not the entire structure of phospholipid, right? So there are other molecules involved. What are these molecules at all? To make this so simple, let's try to number the carbons that we have over here. So we're going to consider this carbon to be our carbon 1, that's to be our carbon 2, and this is going to be our carbon 3. Okay, fine. Now, what is binding to the hydroxyl ions or the hydroxyl groups of the carbon 1 and carbon 2? Binding to carbon 1 and carbon 2 are fatty acid chains, okay, fatty acid molecules. So we have a fatty acid binding to carbon 1 over here, and another fatty acid binding to carbon 2 right over here. Awesome. Now, before we go to knowing the kind of molecule binding to the carbon 3 of our glycerol backbone, there is this concept that you have to cover, okay? Fine. Now, why are we going into this detail, trying to know the typical structure of phospholipid? One reason being that these fatty acid chains that you can see on the board, binding to the carbon 1 and carbon 2 of the glycerol backbone, have something to do about the particular property of the cell membrane structure. Okay? In the cell membrane, later on we realize that the cell membrane is not a static structure. It's not a rigid structure. Okay? It has the ability to flow. Okay? It has the ability to move laterally to displace its protein and carbohydrate contents to the lateral side. So for example, let's consider this to be a cell with its cell membrane. Taking this to be our cell with its cell membrane, okay? This is our cell nucleus, right? Say we place a protein on top of this side of our cell membrane, okay? This is going to be a protein molecule. And we give this protein a little time. By the time you realize, the next time you come and take a look at your cell membrane structure in relation with the protein that you placed over here, you see that the protein has shifted a little bit to this side, meaning that the cell membrane structure is not rigid. As time passes by, it moves, okay? It has that lateral movement. We call this property of cell membrane the fluidity of the cell membrane. Take note of that. What's the name of this property? We call it what? The fluidity of the cell membrane structure. Okay? And this property is affected by a lot of factors. Fine. Now, entering into this detailed concept is going to help us to discover some of these factors. Okay, so one factor that is concerned our phospholipid structure we have on the board right now that affects this special property of cell membrane called fluidity. It's about the length of the fatty acids binding to the hydroxyl groups of our carbon 1 and carbon 2 of our glycerol backbone. Okay? Yes, now how? How does the length of the fatty acids affect the fluidity of the cell membrane? Okay, fine. This is where you keep your rule in mind. And the rule says that increasing the length of the fatty acid chains decreases the fluidity of the cell membrane. Okay, so what are you trying to see? Let's try to put it in the diagram to make it a little bit clearer. So when you consider this cell with its cell membrane plus the protein associated with it to be our cell type A. Okay, so this is going to be our cell type A. And then a cell type B is also going to be placed by its side, whereby what is differentiating between the cell membranes of cell type A and cell type B is their fatty acid content. Okay, so let's consider cell type A cell membrane to have fatty acids binding to the phospholipid structures in there to range from 20 to 24 carbon atoms. Okay, you know that the length of the fatty acids are determined by what? The number of carbons that are confined in the fatty acid molecule. Okay, so in our cell type A cell membrane, the phospholipid structure have their glycerol backbone binding to fatty acids ranging from 20 to 24 carbon atoms. Now, what about the cell type B cell membrane's fatty acids? We're going to consider fatty acids in cell type B cell membrane ranging from 12 to 14 carbon atoms. Okay, yes. So now you can tell me that in these different cell membranes fatty acids, the length of the fatty acids in cell type A, okay, is going to be what? Longer than that of what? The cell type B's fatty acid chains. So because of that, we come back to our rule which says that increasing the length of the fatty acid molecules, okay, in the cell membrane decreases the fluidity of the cell membrane. So now you are going to tell me by yourself that which of the cell membranes will be able to displace its protein to the right faster. Supposing we try to place a protein on top of our cell type B over here, and then we give these cell membranes some time to displace their protein molecules. Which of them will be able to displace its protein faster to the right? You know that the one that will be able to displace its protein faster to the right is going to be the cell membrane with a higher fluidity, right? So now which of them, depending on the rule that I've stated already, and the difference in the carbon atoms confined in the fatty acids binding to the carbon 1 and carbon 2 of the glycerol backbone in the phospholipid structures, which of them will have a higher fluidity? Obviously, it's going to be our cell type B cell membrane. Why? Because in cell type B cell membrane, the fatty acids in there are a little bit shorter. For that matter, it's going to increase the fluidity of the cell membrane in cell type B. Excellent, guys. That is how the length of the various fatty acids binding to the glycerol backbone of the phospholipid structures affect the fluidity of the cell membrane. Now, there is one other thing. With regards to these same fatty acid chains, the level of saturation of the fatty acids also have got something to do with the fluidity of the cell membrane. What are you trying to say this time around? You are saying that the level of saturation, okay, of the fatty acids also affect the fluidity of the cell membrane. Very soon you realize how the fluidity of cell membrane is so important to its function, okay? But for now, in our typical phospholipid structures, we're getting to know certain factors that affect this fluidity. We've already established the fact that 1, the length of the fatty acids affects the fluidity, and we've already explained that. Now 2, we are saying that the level of saturation of the fatty acids also affect the fluidity of the cell membrane. How? What does it mean to say saturation when it comes to fatty acids? Fine. In fatty acids, there are lots of ways we classify them. Fatty acids can be classified in a way concerning whether the fatty acids have double bonds in their structural formula or not. So for some groups of fatty acids whereby there are double bonds in their structural formula, we term them as unsaturated fatty acids. How do we call these types of fatty acids? We call them unsaturated fatty acids. Why? Because they have double bonds in their structure. Okay, Very soon, we're talking about lipid biochemistry, whereby we'll deal with these concepts into detail. But for now, we are saying that in some types of fatty acids, there are some double bonds in their structure formula. We call them what? Unsaturated fatty acids. And therefore, there are other fatty acids whereby there are no double bonds in their structure. So we call them what? Saturated types of fatty acids. So fatty acids can be classified in this manner. Why do we have to know this? Concerning these fatty acids binding to the glycerol backbone, there are some phospholipids whereby the fatty acids in there are all saturated. So you realize that the fatty acid 1 and the fatty acid 2 are all saturated fatty acid types. And then we get some phospholipid structures in the cell membrane whereby we realize that these two fatty acids will tend to be both unsaturated fatty acids. And there are some commonly existing situations whereby you realize that for that particular type of fatty acids, or for those groups of phospholipids in the cell membrane structure, one will be a saturated type and the other will be an unsaturated type of fatty acids. So this means that there are different levels of saturation of phospholipids in the cell membrane structure. And we are asking ourselves right now that how does these different levels of saturation affect the fluidity of the cell membrane structure? Is that okay? Awesome.